We are starting with slide number 39 in the chapter 25 slide set A. The two types of nephrons, remember that a nephron is one unit of the kidney that produces a small amount of urine. There are millions of nephrons, and all of that urine is collected, and sent to the bladder. But each nephron produces a very small, very minute amount of urine. So nephrons that we call cortical nephrons, which is about 85%, are shorter. They're found almost entirely in the cortex. But then we have juxtamedullary nephrons, which we'll spend the most time on. These have these long loops called nephron loops or loops of Henle that travel deep into the medulla. So what their purpose is to produce concentrated urine. So here you can see a picture of, would be a cortical nephron. You see how much shorter it is, and this would be a juxtamedullary nephron. See how it has this long loop that goes deep into the medulla of the kidney. There are two capillary beds associated with each nephron, the glomerulus, of course, and that is part of the renal corpuscle where filtration occurs. And then there are the peritubular capillaries, and they're just capillaries that are associated with the renal tubule. And then the juxtamedullary nephrons have blood capillaries called vasa recta that travel parallel to the nephron loop. All right, for the glomerulus, the capillaries are specialized for filtration. And as far as the arterioles that serve the renal corpuscle, the one that enters the glomerulus is called the afferent arteriole, and the one that leaves is called the efferent arteriole. Sometimes I say afferent and efferent just because it's hard to hear the difference. But afferent is the arteriole that enters the glomerulus, and the efferent is the one that leaves the glomerulus. The vasa recta, again, are parallel to the nephron loops in the juxtamedullary nephrons, and they help to form concentrated urine. So we can blow this up. The glomerulus is inside the Bowman's capsule. Then you see in purple here, you can see the peritubular capillaries. And then in purple over here, associated with the nephron loop, are the vasa recta. Okay, there are 3 cell populations in the juxtaglomerular complex. And we'll look at a picture in a minute, but the juxtaglomerular complex is basically where the DCT comes up through the afferent and efferent arterioles. And you'll just have to see a picture of that to see what I mean. But the cells are called the macula densa. These are tall, closely packed cells of the ascending limb. The ascending limb of the nephron loop, and they contain chemoreceptors that sense the salt concentration of the filtrate. And then you have granular cells, or sometimes they're called juxtaglomerular or JG cells, and they are smooth muscle cells of the arteriole itself. They act as mechanoreceptors, and they sense blood pressure in the afferent arteriole, the one that enters the glomerulus, and they contain secretory granules that contain the enzyme renin. So here's a picture. Good, because it's kind of hard to understand without a picture. Okay, so you can see this portion is the ascending limb of the nephron loop. I think I said it was the DCT, and I apologize for that. It's where it becomes the DCT. but it's technically the ascending limb of the nephron loop. And these cells are macula densa cells. So they're actually cells of the nephron loop that come in contact here with the afferent arteriole and the efferent. So these are macula densa cells, and then these cells are it says extraglomerular mesangial cells. We really won't talk about them, but the granular cells are here and here. So all of this is the juxtaglomerular complex, and what they do is they help to monitor and regulate blood pressure and concentration of the filtrate. All right, section 25.3 is on physiology of the kidney. The kidney produces 180, well, it processes 180 liters of fluid a day, but only forms 1.5 liters of urine because most of that is reabsorbed back into the blood. The kidneys filter the body's entire plasma volume 60 times a day. They consume 20 to 25% of the oxygen used by the body at rest. And filtrate, which is the fluid produced by glomerular filtration, is basically blood plasma minus the proteins that are found in blood plasma, the large proteins that are too large to filter through. Urine is produced from the filtrate. So it's called filtrate before it's called urine. Urine is less than 1% of the original filtrate. If you do this math, you can see 1.5 is less than 1% of 180. Urine contains metabolic waste and unneeded substances, and the necessary needed substances are reabsorbed back into the blood. There are 3 processes involved in urine formation. They are glomerular filtration, which happens in the glomerulus, tubular reabsorption, which primarily happens in the PCT, or proximal convoluted tubule. This is selective reabsorption of 99% of the substances from the filtrate back into the blood, including glucose. All of the glucose that was filtered is going to be put back in the blood. And the third process is called tubular secretion, and mostly this happens in the DCT, or distal convoluted tubule. And that is the selective movement of substances from the blood back to the filtrate to be excreted, with the urine. And this just shows you the 3 processes and where they occur. Filtration occurs in the renal corpuscle. The blood is filtered through the glomerulus, and filtrate is collected in the Bowman's capsule or glomerular capsule. Your tubular reabsorption happens mostly in the PCT, and secretion happens mostly in the DCT. But you can see that reabsorption means from the filtrate back into the blood, and secretion means from the blood back into the filtrate, or the urine. The filtration membrane itself is really the portion of the glomerulus, or the blood capillaries, that are in contact with the Bowman's capsule, the portion that the blood is filtered through. So there are 3 layers. First of all, the fenestrated endothelium of the glomerular capillaries. Now, endothelium is just the name for the lining that forms the capillaries. They're simple squamous epithelial tissue is what it is. It's just a single layer of epithelial cells, but they're fenestrated. They have fenestrations. They have slits in them. There is a basement membrane that's really fused between the membrane that forms the capillaries and the membrane that forms the Bowman's capsule lining. So you have 2 fused basement membranes, one from the nephron and one from the blood capillaries. Then you have the foot processes of the podocytes with their filtration slits that are formed from the nephron portion, the Bowman's capsule. All right, and here you can see the Bowman's capsule. Okay, it's got two layers. It's kind of like a serous membrane in that way. It's got an outer layer called the parietal layer, and then it's got a layer that touches the capillaries. And that's what forms, these are podocytes that form those foot processes. So that is the visceral layer that actually contacts the capillaries. So the filtration membrane is formed from the lining of the capillaries of the glomerulus and the lining of the visceral layer of the Bowman's capsule. So blood is filtered through using the pressure of your blood pressure. Blood is filtered through the glomerulus into the Bowman's capsule. And so that's what we call the filtration membrane. Again, it's just the cells of the capillaries, the basement membrane of both the capillaries and the Bowman's capsule, and then those podocytes. This is a podocyte here. It's a cell. You can tell it's a cell. It has a nucleus, and it's got these extensions, and they form filtration slits that allow the blood plasma to be filtered through into the Bowman's capsule. And this is another diagram of that. Okay, so the filtration membrane allows molecules smaller than 3 nanometers to pass, and what we need to know about that is water, glucose, amino acids, and nitrogenous waste like urea. And then plasma proteins stay in the blood and prevent the loss of all water to the capsular space, the Bowman's capsule. The proteins in the filtrate, if you do have proteins in the filtrate, that indicates that the membrane is damaged. The filtration membrane is damaged because proteins are typically too large to pass through that membrane. Now, the pressures that promote filtration are hydrostatic pressure, which is really blood pressure, okay? And there's this whole talk about pressures, but we're really going to be more concerned with the net filtration pressure. And net filtration pressure comes from the forward pressure of your blood. This is the hydrostatic pressure, or the blood pressure has to be greater than the pressures opposing it. And it is. So in healthy kidneys. All right, GFR means glomerular filtration rate, and this is a measurement of kidney function. It's the volume of filtrate formed per minute by both kidneys. In a normal kidney, or in normal kidneys, that is 120 to 125 milliliters per minute. GFR is directly proportional to net filtration pressure, total surface area, and the permeability of the filtration membrane. So GFR basically is directly proportional to blood pressure, and where we get into there is the relationship between the cardiovascular system and the urinary system, because there's a strong relationship. If you've ever known someone, especially if I know a lot of you work in nursing homes. A lot of my students end up working in nursing homes, at least for a little while. If you've ever been there, during the death process, if their kidneys stop functioning, then their blood pressure is gonna drop. If their blood pressure drops, their kidneys stop functioning. It all kind of happens together. If a person has kidney disease, they tend to have cardiovascular disease. If they have cardiovascular disease, it tends to also correlate with kidney disease, because both of these systems are dependent on one another. So intrinsic controls of regulating glomerular filtration. The intrinsic controls is called autoregulation, renal autoregulation, and there's two types: the myogenic mechanism and the tubuloglomerular feedback mechanism. So this is how we regulate filtration rate. First of all, this is really a simple thing. The myogenic mechanism is I'm trying to think of like how to say it a simple way, because it's very, very simple in theory, is higher blood pressure causes muscle to stretch. And the more stretch there is in the smooth muscles lining the afferent arterioles, the more they constrict. It's like the higher the blood pressure is going to cause those afferent arterioles to constrict, which will then lower the blood pressure. And too low blood pressure, which means low GFR, will cause those afferent arterioles to dilate. So that's the myogenic mechanism. And the tubuloglomerular feedback mechanism is controlled by the macula densa cells. Let's go back and look at those. The macula densa cells in our picture somewhere. Here we go. Remember, those are the cells that form the nephron loop, the ascending portion of the nephron loop, as it comes up through the afferent and the efferent arterioles. And these cells are called macula densa cells. All right, find where we were. There we go. Those macula densa cells are able to sense the sodium, the salt concentration in the filtrate. And they will respond to that. So higher salt concentration correlates with higher blood pressure and higher blood, anyway, they're going to detect higher sodium salt concentration, and I'm trying to think of the best way to explain it. Okay, well, let's just look at it. I didn't highlight it in yellow, but I'm going to read it anyway. If your filtration rate increases, this leads to decreased reabsorption time. So the faster that filtration is, the less materials can be reabsorbed back into the blood. And that will lead to high salt levels in the filtrate, which will be detected by the macula densa cells. That will cause the constriction of the afferent arteriole, which will lower the filtration rate, and that will give more time for sodium and salt reabsorption. And it's opposite when the GFR is decreased. So hopefully you're noticing that it's important for the GFR to be exactly where it needs to be. Filtration rate doesn't need to be too high, because then there's not enough time for enough reabsorption back into the blood of necessary materials. But if the GFR is too low, then you won't have enough pressure to filter. There's not enough pressure to filter the materials. So again, too low and too high are both bad, you know. So we're going to start with, this is slide number 71. And we're going to start with slide number 71 in the next recording, because I've gone past my time.